about some important experiments that led us to understanding that DNA is a carrier of our genetic information, right, and not proteins. Today we're going to start talking a little bit of history about how we determine the structure of the molecule and kind of review some important things about the structure of DNA. So, you might be familiar with the names of the scientists who first came up with the first model of what we know DNA structure to be now. They're Watson and Crick. So those are the two scientists, one American, one English, who kind of worked together and came up with that first double helix model of DNA. It shows the two strands wrapped around each other. You're probably really familiar with. However, there are also some other scientists that were working on this question at the time, maybe most notably Rosalind Franklin, who contributed a lot to the model, but was not really recognized at the time at all. And the other two gentlemen who were working with her at the same institution. So those are kind of the competitive group that was working along and with Watson and Crick. So we'll talk a little bit about Franklin's contribution before we talk about the model itself. So just kind of a timeline. When you see these dates in the corner, this blue date is like the date of the experiment or the date that the model was formed, and then the red is when they received the Nobel Prize for it. So that's what those dates are up there in the corner. You're not going to have to memorize those, just kind of just for your own information. So, Rosalind Franklin, as a woman at the time, faced a lot of discrimination, and her contribution to the structure was from these photos, this X-ray crystallography, which you basically have to take a whole class in order to really read these images. But the reason why this image was so vitally important is because it showed us that DNA was in fact a double helix, that it was two strands that were wrapped around each other. And this was pretty paramount information to help us figure out what DNA structure was. So that was Rosalind Franklin's contribution to this process. And without it, Watson and Crick really wouldn't have figured out the model itself. And so we're going to take a look at their paper. And in that paper, nowhere do they reference the fact that they got this information from her. So here's Watson and Crick. Actually, go ahead and get your Chromebooks out. I gotta link this in the agenda for today. Okay, and I'm just not putting the link in, so you might have to refresh. But if you go to today's agenda, it's not very often we look at primary literature. So it's not a very long article as far as primary literature goes. So. Want to give you some time to kind of read through that. And obviously, some of it's pretty technical, so see what you can get out of it. Take some time. Let me get you time to actually go through the Oh, it should be in the agenda. Get it. Might have to refresh it. This is what they actually published back in 1953. It is. So basically at the beginning here, they're talking about the other ideas for DNA, which is that instead of a double helix, it's a triple helix, and they talk about reasons why they don't think that's right. So first of all, if you have a bunch of negatively charged phosphates next to each other, what are they chemically going to want to do? Repel. So instead of the phosphates being on the inside of the axis, they're saying they're on the outside. Okay. And if you take a look at figure 1, they say the two ribbons symbolize the phosphate sugar chains, the backbones, and then the horizontal rods are showing the bases. And again, they really only knew for sure that it was a double helix because of Rosalind Franklin's work. And at no point in this do they even reference that that's where they came up with it. They also talk a little bit about the base pairs, how they came up with the idea and the chemical pairing between adenine and thymine, and guanine and cytosine. So purine with the pyrimidine and purine with the pyrimidine. So those terms might be kind of more familiar for you than some of the other ones. So they do actually mention that there was some published X-ray data, but again, they don't really mention that they got. I guess they do mention here at the bottom, but she didn't really get any the full recognition for her contribution to the model. So a lot of people think she should have gotten part of the Nobel Prize along with them for this experiment. So anyway, just not gonna test you on anything specifically in this paper, but kind of give you an example of some primary work and what that first publication about DNA structure looks like. All right, we'll go back to the PowerPoint now. So I have two diagrams of a nucleotide up on the board with your group members. Can you identify those parts? Oh, they're both nucleotides.
Well, I see that.
Okay. So they are slightly different from each other, but both of these contain a phosphate group that's then bonded to a sugar. In DNA, that sugar is deoxyribose. In RNA, that sugar is ribose. That's where the names come from, right? DNA is deoxyribonucleic acid. RNA is ribonucleic acid. That sugar is then connected to a nitrogenous base. Yeah, and some bases are made up of one ring, some bases are made up of two rings, right? So this is a pyrimidine and this is a purine. Okay. They're called nitrogenous bases because they contain nitrogen. Okay, so these groups are polar. This ketone here with oxygen and these amine groups hanging off are going to be polar. And so when you have two bases next to each other, what type of bonds are going to form between them? Hydrogen bonds, right? So on Monday we'll do a modeling activity where we're going to draw out a specific DNA molecule. And when you draw those nucleotides, you need to indicate that there's a phosphate group, a sugar group, sugar, and a base. Questions on nucleotide structure before we move on? Okay, 2, base pairing in DNA. With your group, really quickly review how do bases pair? Okay. So, and looking back to Chargaff's experiment, right? We know that the percentage of A is roughly equal to T, so A and T are going to pair together, and the percentage of C is roughly equivalent to G because C and G pair together, right? So we have adenine and thymine, and we have guanine and cytosine. They also want to point out that in each of these pairs we have a 2-ring base, purine, and a 1-ring base, a pyrimidine. So if we think about the width of DNA, the reason why those backbones run parallel to each other is because you always have a similar distance between your pairs, right? You have a big base and a little base, big base and a little base. So that molecule is going to be straight. If it wasn't that way, then it would be like kind of curvy, right? So if you have 2 big bases versus 2 little bases, that those strands, the backbones would be parallel to each other. Right, here's the next question. What type of bond is holding the bases together? What type of bond is holding the sugar phosphate backbone together?
What? Is it yeah, I think so.
Okay. All right. So what type of bonds holding the bases together? Hydrogen bonds. Remember we mentioned this already. We have these polar functional groups that are attached, so they're going to form hydrogen bonds with each other. Notice that T and A form 2 hydrogen bonds, and guanine and cytosine form 3. So and that again goes with the base pairing, like they line up to form hydrogen bonds only with the other base pair. So C and D can only really form good hydrogen bonds with each other, and A and T can only form good hydrogen bonds with each other. Hydrogen bonds themselves are relatively weak, right? Which means it's fairly easy to separate the two strands from each other. All of these bonds in the backbone, the sugar phosphate backbones, are going to be, well, they're diester bonds, but they're covalent. Okay, so these are covalent bonds. Relative to hydrogen bonds, covalent bonds are much stronger, right? Because they're physically sharing electrons between the nuclei. So when we replicate our DNA, these backbones are going to stick together, but we're going to separate it between the bases, right? So we can kind of unzip the DNA molecule. We'll talk more about replication in the next section. Questions on that? Okay, so here again, it's just kind of an overview of the structure. A lot of times we see it look like a ladder, but just kind of keep in mind that it's actually a helix. Technically, it's a right-handed helix. So what that means is if you put your thumbs up like this, and you see the way that your fingers are curling, DNA is a right-handed helix. So the molecule is curling in that direction as opposed to curling in that direction. So if you kind of see that curling around the axis. Okay, this is probably something that you haven't ever learned, but when we take a look at that sugar, in this case we're looking at an RNA nucleotide ribose, but this is true for the oxyribose too. The carbons are numbered, okay, starting with their attachment to the base. So there's the first carbon, and then they go around in a circular manner there. 1, 2, 3, 4, 5. So I think it's helpful to remember that the 5' carbon is attached to the phosphate. So there's a little alliteration there. 5' is attached to the phosphate. So that helps me to remember which carbon is the 5' carbon. The other important carbon is the 3' carbon. We'll talk about why in just a second. So we'll kind of explain why this bond forms the way it forms when we talk about replication. But when we add nucleotides to each other, they're bonded from that 5' carbon, from that phosphate group attached to the 5' carbon to the 3' carbon. So if we were going to add another nucleotide to the bottom here, the phosphate group would then attach to the 3', and that's going to continue. So when we add nucleotides, we always add the phosphate group to the 3' carbon. So we're always adding 5' to 3'. Yeah, we'll talk a little bit about why that happens next section. And you might have noticed those numbers in the Watson and Crick article. So that gives some direction to a strand of nucleotides. So whatever side of the strand is ending with the phosphate group free is the 5' end. So again, phosphate and 5, there's a little alliteration there. Okay, so wherever the free phosphate group is, that's the 5' end of the strand. Wherever the carbon is free, the 3' carbon is free, that's the 3' end of the strand. Okay, so 5', 3'. So that'll be especially important when we get to replication again. The last important thing we're going to talk about here has to do with the fact that DNA strands are anti-parallel. So first of all, parallel means they're equally spaced apart, right? Anti means they're going in opposite directions. Okay. So those paired strands are connected by their base pairs run in opposite directions. So talk about this with your group members. If you take a look at that diagram there, can you find the 5' and 3' ends for each one of those strands?
And then the 3 is on the bottom left. I don't get it.
So these the 3 phosphate ends, again, those are the 5' ends. And the 3 carbon end, those are the 3' ends. Okay, so again, they're parallel, but they run in opposite directions. The 5', 3' here, and 3' to 5' on this side. Okay, and you can actually see they're kind of showing you that the molecule is like reversed, right? So our phosphate and our base letters are upside down. So, questions on that? Yeah.
The 3, why would it not be 5? There's like a 3 like right there. Or this.
Well, this is the 3' carbon here. So you have 1', 2', 4', 5'. So this is the 3' end. This is the 5' end. And they're opposite here, right? Here the phosphate group is down here. It's the 5', that's the 3'.
Below that, where's the big note below that? This says 3'. Why wouldn't it be modulo 5 and 1?
Well, if we add another phosphate, if we add another nucleotide here, then it would go phosphate 3'. Does that make sense? So this is the end of the strand. There's like no nucleotide underneath it. Okay, the 3' carbon is free. Okay. All right. So I meant to have another activity for you, but it's not ready. So quick check. There's quick checkup. Try and finish that before you leave. And then next section of your study guide, you should be able to fill out. Okay, so quick check through the next section of your study guide. But it's been a long week.
It's been a long day.
Already, so serious.
We got the back of the page.
That was pretty good. Exactly. Look at our life. What?
I don't know. What? We're 60.
Realize innocent what you like to go to sleep. Go to sleep.
I don't know if I. I don't. I mean, so
I'm not very good at that. But I'm very
practice something you can't remember. Yeah. I think Nancy.
Why would I want to see hydrogen? I don't know why. But normally when I do that, why do people
Oh, two hydrogen.
I know it's me, but I don't really Yeah.
You can. I haven't tweeted
One to six.
Isn't it, it's like, I don't know what I
I've been 25 since we control like. Now it's at 3.227. Now I did it again. I have to do it again, because I hold it too. I missed four. This isn't funny, bro. I'm gonna do it again. What's that?
And Josh
Looking at these steakhouses. Good, the size are ginormous. Yeah, let's hold the shit the size. That's why I read this trip online.
Number 3.
This dude gave us a 125 off. We did it. It was his coworker.
You can maybe switch that into being yeah, just do something maybe that you put in the box. But I would somehow make sure you know that like the sugar is central. Maybe just
I can't. Why now? All three. Plus my house is my house. The living room is 2 stories. Why the hell is I don't know why. I'm not dang, this place is like, no. Okay, so but no, the living room is a giant box that's 2 stories high. Why do you need 2 story living room? What's wrong with it? And it's so loud. There's a massive fan out there. The fan is 2 stories high. No slippery. One time when I was oh god. I'm not done yet. Hey, let's see. The height of this building is the height of this building. This is the fan is all the way up. How do you put that?
I'm not just.
Yeah, I got one.
I'm going. I can buy them.
Yeah, there is one down.
Yeah, you know how do like the music uncle was like like run down like little house where it comes like down to it costs $1 million. Oh my god, California house prices. Toronto's house prices.
And then like laugh.
What way house prices are
I mean, I like the people.
Hey. Okay, glasses. Then take off your tie. They give you
The human race is an invasive species.
Oh my God.
doesn't keep your business.
No, yeah.
I am a close friend of mine on the first two. It's not fancy. You can't pull that. I'm really trying to find it. Nothing to watch. Yeah, I was just chopping it down. Well, I think he like he dressed you. Black sock. Wait, these are all dresses. He has the same police here. The stock image has the same. That's okay. He has a watch. He doesn't have the time. That's
So, all of these,
I'll take it.
I got 100. And then these two things.
is a Twitter. You go on Twitter. This is Twitter. This is Twitter. This is Twitter. This is you. So similar. What?
I don't want Bill Gates in my friendship.
Our time 53 million followers, but only getting 4.2K artists in the day. Rock. 128. 10 hours on a Sam. 128. Yes, it's not loud. Oh, dude. No, because it's happening. No. Yeah, I've never treated. No, I feel like he has the big one. I actually don't have to do it. He's a big one. I have to look how to
Yeah, I know.
No, I'm good.
It doesn't get that long. I don't like the brown. The best decision ever. Yeah, but it takes too long.
Go on.
people need to come to the attendance office. Connor Tchaikovsky, Olivia Eiler, Sydney Ruth, Lauren Phillips, and Bailey Stahl.
Super long, super far.
So technically only 10 points.
Anything goes better. Okay, great.